that's deals that help you. And it's just like this. You gotta do it. You need to a bit of fun.
International guest speaker.
Being busy.
Matthew, right on.
So I had to go through it. I went through 2 sessions.
Where's Kim first? Since I've been out talking, I've been you showed
Small one.
So we're in the
What?
nerve.
Right here. Kim, I don't mean to disturb you, but
Grabbing something, to do something, we know there's something in your situation is up here. And also, don't.
yeah, then we gotta get it. 30 milliseconds. We'll be right back.
Right through here, so in fact, when I ring things out, I just hold things straight with this and I'll do my ringing with my, so far I haven't had much trouble with this one, so I don't need. But that's what, there's a bunch of little things that all of a sudden that you.
Jason Ray.
Maybe there've been some changes. It's like a torque on your wrist. There have been changes. Torque doesn't like it.
Yeah, but he's stuck in Hawaii.
I'm gonna check. All right.
So we.
Oh, it's one of those. He's gonna get one. That's not like words.
It's fine, it's lemon.
It's lemon. Yeah. Doesn't matter. We'll go back to all right. Well, good afternoon, everybody, and welcome to our Eco Lunch and Learn. It's the first one for 2026, and we're actually in our 8th season, so that's pretty exciting. It's been an interesting ride, hasn't it, Kim? We started it from a conversation outside of my house, I think it was, and it's grown into this. So we're very excited to have you and see how it has grown over the last 8 years. Just to kind of let you know, Best and Bermuda College were co-hosts for this event, and we try to showcase local experts in the environmental area. And we're very fortunate to have Dr. Sawall.
Yeah.
Did I say it?
Almost perfect.
No, it's not quite, but almost. So I just give you a little quick background of Dr. Sawall and what she's all about. So Yvonne is a marine benthic ecologist with a focus on coral reefs, combining physiology, ecology, and oceanography. She investigates how organisms and communities interact with their environment under current and future conditions. This work includes natural processes such as seasonal dynamics and the impact on hurricanes on reefs, as well as human-caused stressors, including pollution and rising ocean temperatures. Over the past several years, Yvonne has focused in particular on the effects of ocean warming and has begun exploring novel interventions to mitigate heat stress-related coral mortality. Dr. Sawall received her PhD in 2011, and I'm not going to say this right, from the Leibniz Center?
Leibniz Center of Tropical Marine Ecology.
Okay,
yes.
Wonderful, in Germany. She was a postdoctoral researcher, followed by a position as a research scientist before coming to Bermuda in 2016. During this time, she conducted an extended research expedition to Thailand, Indonesia, and Saudi Arabia. In Bermuda, she joined BIOS as a postdoctoral researcher, working with Eric Hochberg and participating in a series of NASA-funded expeditions to Hawaii, Australia, and Palau. In 2020, she became a faculty member at BIOS, and in 2022, she was appointed assistant professor at ASU. Over the past several years, her work has focused on Bermuda's coral reefs, and she has been awarded multiple competitive research grants that enable her and her team to advance our understanding of reef ecology in Bermuda and coral thermal tolerance more broadly. Dr. Sawall has also worked closely with the Department of Environment and Natural Resources on issues related to seagrass ecology, and she is deeply involved in student mentorship, teaching, and public outreach at BIOS and ASU. We are so honored to have her. I'm very excited to hear what she has to say. Thank you and welcome.
Thank you for this nice introduction. I actually prepared a slide to introduce myself, but I can skip that one. That's great. How do I get to my presentation now?
Oh, shoot. I thought Diaco had that.
Is that
Oh, there we go.
okay? Easy, and I'm gonna go. Okay, that's how I go forward. Do I have a pointer?
I'm sorry, we're not that.
Okay.
I'm sorry.
Okay. Okay. Yeah, so welcome. And so this presentation today will be about heat stress on corals and coral reefs. So, and what we can do perhaps to help corals to survive a warmer ocean. Yeah, quickly, just what I do in my lab. I'm going to go through this real quick because we just talked about it. The major thing I do at the moment in my lab, the Marine Benthic Ecology and Ecophysiology lab, is looking at coral thermotolerance and global warming mitigation strategies. Secondly, I work on organismal metabolism, meaning like photosynthesis, respiration, calcification of organisms, and what is driving these processes and reefs. And then I do a bit of seagrass ecology as well, although I do not have any active grants there at the moment. I'm also now slowly getting interested in marine carbon dioxide removal work. That's something that's really an advancing topic, and some of the facilities at BIOS we have are ideal for that kind of research as well. Okay, so layout of the presentation today, I will give an introduction, a bit like lecture style introduction on coral reefs and coral biology in general, and then about the problem of warming oceans and what it does to the corals. And then I will go and provide some examples of my own research. I will talk about some experiments I've conducted in the Red Sea, where corals are exposed to exceptionally high temperatures and how those corals are adjusted. And then, of course, I'll talk about some of the research I do currently in Bermuda. And here we really focus on like how different species react to thermal stress, and then what can we do to help corals to survive warming oceans. Okay, so very general, so we're all getting on the same page on what corals are, what coral reefs are. Here are a few pictures, an aerial picture of a coral reef. I guess everyone has seen that here, who has been flying, out of Bermuda. Then if we dive into the water, of course, we see, many different coral colonies. They form a coral community. And then if we look into zoom in even further and we look at the coral colony, you can see that we have, that the colony actually exists of a lot of individual polyps. And so these polyps are all connected to each other. They can communicate with each other. They exchange nutrients and so on. So they're forming like one kind of superorganism with all these individual polyps. And then inside the polyps, actually, we do have zooxanthellae, which are unicellular algae. So they live inside the tissue of the coral. Here's a cross-section of a coral polyp. So we can see here a stomach, right? So there's where digestion is happening. We have a mouth. We have tentacles that are surrounding the mouth, so they're feeding and particles in the water. And then here, zooming in further, we can see inside the tissue, like inside the cell of the coral animal, we actually have plant cells sitting inside, and those are the zooxanthellae. And they're super important for corals because they're relying on them, and there's a lot of exchange of material between the zooxanthellae and the hosts. And we call this symbiosis, so they're kind of depending on each other. Here you can see a zooxanthellae, which is, through photosynthesis, is producing sugars, lipids, and amino acids, which they then provide to the coral host. So that's a very important source of energy for the coral host. And then the coral host, in return, it's respiring, and during respiration, it creates a lot of CO2, which is then taken up again by the zooxanthellae. The host also provides a lot of nutrients. So when I talk about the host, I mean, that's the coral animal, right? So, and then as the coral host breaks down organic compounds, it also releases a lot of nutrients like ammonium, nitrate, phosphate, which is then also, again, used by the zooxanthellae again. So there's a really tight recycling. And why this is so important is, the reason for that is because a lot of corals we find in very clear water. There's not a lot of food for the coral to feed. So they are relying on the zooxanthellae to provide energy through photosynthesis. Okay, a few facts about coral reefs. Here are actually a couple of pictures that are quite contrasting. One is from the Indo-Pacific at the top, and the other one might look very familiar. This is how the reefs look in Bermuda. What you can see is the diversity definitely varies. As there's a lower diversity in Bermuda. Yeah, it's actually one of the coral reefs here is the lowest diversity on Earth. We only have around 20 hard coral species. But the point here is it still provides this habitat for lots of other organisms. It creates the really nice three-dimensional structures that creates a home for many fish and other invertebrates. So it has the same function, and it doesn't matter now if we have, few coral species in Bermuda or lots of coral species in the Pacific. Coral reefs are also called the rainforest of the sea, and because it is so diverse and it creates a habitat for many different organisms. About 25% of all marine species are found in coral reefs, even though the global area of coral reefs is actually comparatively small. It's only about 2/3 of the size of California, just to put it in perspective. And coral reefs also have a really high ecological and economic value. So it's estimated about $36 billion per year in terms of economic value. And what's really driving the economic value is that coral reefs are really good in buffering surges from storms and hurricanes. So it's protecting coastlines from storm damage and properties from storm damage. It has a high value in terms of tourism, in the tourism industry, but also fishing. Okay, so now there are some major threats to coral reefs. There are local threats as well as global threats. Local threats are eutrophication, pollution, overfishing, physical destruction. Global threats are ocean warming and ocean acidification. And it has been estimated that about 30 to 50% of all coral reef area has been lost since the 1980s already. So this is quite a lot. Bermuda reefs are actually in a really good state if you compare it to many other places around the world, in particular in two places in the Caribbean. So luckily we are, in a fairly good position still. Ocean warming is the major threat globally to coral reefs. And so that's why we will be talking about this a bit more today. So you might be familiar with these figures. This is showing the trend in sea surface temperature, predicted increase in temperature over the next several decades. And in red, you can see worst case scenario. Blue is the best case scenario. We will probably end up somewhere in between, but hopefully closer to the blue scenario. And also the amount of marine heat wave days is increasing. So you can see on the y-axis, the multiplication factor. So it has already increased by about 5 times the amount of heat wave days, and it's also predicted to increase further. An example of Bermuda, so this is some data I took from the NOAA Coral Reef Watch website. So this is the last 40 years, the sea surface temperature in Bermuda. You can see, of course, it's a lot of up and down. So these are the seasonal variations, winter, summer. But if you then put a regression line through it, you can see the average temperature has been increasing over the last 40 years by a bit more than 1 degree Celsius, which is in Fahrenheit, 2.2 something, 2 degrees Fahrenheit. More than that, 2.2. What's the conversion Celsius in Fahrenheit? 1.8. 1.8? Yeah, okay, anyway, about 2 degrees Fahrenheit. So it has been increasing here as well. And to understand a little bit the impact it has on corals, we kind of need to understand first what's kind of normal for corals. So the normal, maximum mean monthly sea surface temperature in Bermuda has been in the past about 27.3 degrees Celsius. And that's this blue line here. And that's kind of what corals are adapted to here. And then now temperatures become stressful for them if we add another degree of Celsius. That would be about 28.3 degrees Celsius. So that's kind of when corals start to be stressed out. And if this temperature is there for too long, they may actually have some serious consequences and impacts. And I'll tell you about the consequences in a minute too. 2025, last year, was actually one of the warmest years here from ocean temperature. You can see here it spiked, but it also stayed warm for several weeks. And I have actually gotten a lot of emails from NOAA from this website and asking like, how do the reefs look like? Do you think it's bleaching? Keep us updated. Luckily, it's not has been that bad in Bermuda yet still, but I also think that this maximum mean monthly temperature of 27.3 is actually a little low. I think it should be higher. It should be more like 28. Anyway, to a bit more about heat stress. So Bermuda hasn't been impacted that much yet. So I want to give you an example of an area that has been heavily impacted by marine heat waves. And there is the Caribbean in 2023, which was by far the worst marine heat wave that I've seen so far. And so here you can see, so that's the Caribbean in gray, that's the land mass, right? And everything that has some color to it is the ocean. And so the more red or more purple, the more heat stress has been accumulated over time already. So the scale here we're looking at, it's called degree heating weeks. And it's just a metric people have come up with to describe the cumulative heat stress over a period of 12 weeks. So you can see here that, for example, the Florida Keys are already pretty hot here, and some other areas as well. So that was in July. Now we go to August, like a month later, and you can see it's becoming much more red, much more, heat accumulated. Then we have September, and then we have October. And this is really like in October now, we can see lots of areas in the Caribbean have up to like 20-degree heating weeks, which has never been recorded before in this area. And it had really devastating impacts on the reefs, many places. Just to give you an impression of what I've actually talked about, like what kind of impact those heat waves have. So you can see here an example of a healthy reef on the left. Yeah. Bleached, and bleach means now they're losing their zooxanthellae, and I'll talk about this more in a second as well. And then on the right, you can see a dead coral community. So that's after a heat wave in 2015 in American Samoa. And a couple more examples of a bleached reef, and then also a dead reef, essentially. So while the structure on the dead reef is still there, it's still providing a habitat. It's only a matter of time when it starts falling apart. So that's kind of right after a devastating heat wave. Even sea anemones can actually bleach because they also have zooxanthellae. In Bermuda last year, we took a few pictures of bleaching corals. You may have seen that the fire corals in particular turning white sometimes in the summer here. You can see lots of other corals are still fairly, keep their color and look okay. So the top three pictures are from fire corals. And then the bottom left here, you can now see a brain coral, which is also fairly white. And then just to make you aware of it, not every time you go into a reef and see a white patch, it's bleaching. On the right picture here, this is actually a coral disease, and you can kind of distinguish it because it's a patch, and you kind of see how this disease is eating away the tissue. If it's bleaching, typically you have a more homogeneous change of color, of the entire colony. So what is bleaching? So bleaching is, as I said, is the loss of zooxanthellae, so the unicellular algae. And the reason why this is happening is something's wrong with the photosynthetic machinery inside the zooxanthellae. So I'm going to give you a little introduction. Most of you probably are fairly familiar with photosynthesis, but just to kind of give a quick reminder. So we have in photosynthesis, we have the light reaction and the dark reaction, right? So in the light reaction, we have the light is being absorbed by the chloroplast or by the thylakoid, and it's basically catching the energy, and then it uses the energy to split water into oxygen and protons and electrons. And the electrons are then the ones that are transported from one protein to the next down the electron transport chain until it's getting onto the NADPH. And this is like the, oops, where's the mouse? I don't see it. Okay, here. And that's like an energy carrier inside the cells. And this is then used in the dark reaction now, where CO2 is being fixed into sugars. And so that's kind of, and you can see what the main point here is really, you can see that all this is connected. The light reaction, the dark reaction are connected, and there's some recycling of products going on. And so now if we have heat stress, what happens is that some of the proteins or enzymes that are working in this machinery are starting to break down. And that means there's some interruption in this, recycling process here. And what happens next is that the electrons that are coming from the water that is split by the photons of the light becoming too much, and they're not processed properly anymore. So there's an overflow of electrons essentially. And then those electrons then start to react with the oxygen, forming oxygen radicals. We all know oxygen radicals are bad. They're like attacking DNA, they're attacking, cell walls, cell membranes. So there are some mechanisms in place the corals have to kind of fight against that and neutralize oxygen radicals. But if it's becoming too much, then it's becoming really damaging, not just for the zooxanthellae, but also for the coral host, because it leaches out of the cells. And so if there's a point then where there's too much damage being caused by the zooxanthellae, the coral animal says, you know what, you're doing more damage than good, I'm going to kick you out. And that's kind of the bleaching process. And so there are like different ways corals can, combat this process. So very early, they can actually, so when the enzymes start to break down, there are certain other proteins, like the so-called heat shock proteins, that can actually help to prevent the damage or repair the damage. So now you can already think of like some corals might probably be better in doing this than others. That also explains why maybe some corals are more resistant against heat stress than others. Another process is, like I said, there's too many electrons in the system because the light is not, the light is still there. So it's still like shooting all the photons onto the corals. And so there's this excess of electrons. One way of fighting against that is to increase photoprotective pigments inside the coral. So then the coral is able to neutralize some of the excess light before it becomes damaging. Third is okay, now we do have oxygen radicals. So what do we do with those? So there are a lot of antioxidants, like human cells have it, but also, coral cells have it too, that can also then reduce the amount of oxygen radicals. And then lastly, and we know that already, so if none of these mechanisms work properly, the last reserve is really to kind of get rid of the zooxanthellae. And in order to be more tolerant to heat during the next heat wave, some corals are able to actually acquire more resilient zooxanthellae species. So even though, those little unicellular algal cells, they all look kind of the same, they all look brownish, brown, and they're tiny, there are actually many, many different species that's sitting inside the coral host. Okay, so that was the lecture part. So now we're all on the same page about what bleaching is and why it's such a big concern as well. So I want to talk now a little bit about some of the research I've been doing. first of all in the Red Sea, and the Red Sea is here between, Africa and the Arabian Peninsula. And what's really special about this area is that it has really, really high temperatures in the southern part of the Red Sea. These are kind of the hottest temperature where complex coral reefs and diverse coral reefs still exist. So I had a bunch of collaborators there for this work. So that was actually my first postdoc and the work I've done there. So the temperature gradient of the Red Sea, so in the north they have 21 to 28. So that's about actually what we have in Bermuda, quite comparable. Also the latitude is quite comparable. And then, but in the south now we have 28 to 33 degrees. And so the point of this project was, or the objective was to gain a first understanding of the mechanisms underlying high thermal tolerance in the Red Sea. So we went out and first the study area again. So we picked like 6 sites along the Red Sea, along the Saudi Arabian Red Sea coast. And so just to give you an idea, so this is about 2,000 kilometers apart. So it's quite a distance. And then we deployed hobo loggers, like temperature loggers on all these sites. So we know the temperature over a full annual cycle. That's what you can see on the right here. So number 1, so the different curves, right, they from the different sites. So number 1 is the most northern side. Number 6 is the most southern side. And you can see number 5 and 6 are really, they have temperatures above 30 degrees for half of the year. So it's really, really warm. And then we went out and did 2 expeditions. One was in the end of summer, one was the end of spring or winter. So we kind of have the hottest and the coldest period of time. And we did a bunch of different measurements. But we went out and collected just a number of different corals to get an understanding. Do the corals in the north function? Do they have the same genetic makeup than the corals in the south? So are we actually talking about the same population, or are we actually talking about different populations? And we found here, if we just look at the coral animal part first, we can see here from the north to the south, the colors don't really change, which means it's all the same population and we all have the same genotypes here, except in the very south where we do have some different coral genotypes. So we all say it's all the same species, but they do vary a little bit genetically. So that means that there is a high genetic mixing within the Red Sea. So really like once the corals reproduce and release the larvae or their gametes into the water, they actually do disperse quite far and they're really mixing. So it means like, it doesn't matter if they live in the colder region or the warmer region, they're kind of all the same. So that was one thing. And then the other thing was, of course, looking at the zooxanthellae, like how do they look like? And we can see there's a bit more separation between north and south. So we can see in particular in the north, there are lots of different colors here, so meaning lots of different species, zooxanthellae species and genotypes. And then if we go further south, here it's more homogeneous, and then in the south again, it's different. So we can see here a little bit of, or we can kind of guess already there's some local adaptation to the different temperature regimes just by having different zooxanthellae types or species. And then just to complete it, I'm not going to talk about bacteria further, but of course, every coral also has lots of bacteria living on top and inside. And so we also looked at how do the bacterial communities differ between the different regions. And we can see a clear shift here from the north to the south. And they also, bacteria can be good or bad, right? So they can be good. They can also help, help with nutrient cycling. They can also help with certain stress events or combat diseases, but they can, of course, also do the opposite and be pathogenic if environmental conditions change. Okay, so that's kind of the background. And now we look at the physiology of the coral. So we went out and did a whole bunch of different measurements. Here we did some incubations where we put the coral inside the chamber, and then each chamber has an oxygen sensor connected to it, and so we can measure the change in oxygen over time, which is then used to calculate photosynthetic rates or respiration rates if it's in the dark, and also we can measure calcification rates. We did. And we also then took actually samples of all the corals and also did some other analysis, like we look at the photopigments, we look at the protein content and all kinds of different things. And so here's some of the key results. So looking at the calcification first, so calcification is just a fancy word for growth, right? We can see, so now first, let me explain this figure first, how to read it. So here we have the different sites. Again, number 1, the most northern, number 6, the most southern reef. And then we have in red, measurements taken in the summer, like at the hottest time of the year, and then in blue in the winter, coldest time of the year. So we can clearly see here already, a change from north to south. And also that the seasonal variability is highest in the north and is lowest in the south. But also we can see a reverse pattern here. So actually corals grow better in the winter here in the south than in the summer. So we kind of can already guess there's a relationship with temperature. So if we do actually plot this against temperature, we can see a very nice relationship. We can see that here, as temperature increases, calcification increases until it reaches the optimum here at around 28, 29 degrees Celsius, and then kind of starts to drop again. So again, these numbers represent the different sites, and the colors represent just a different season. This result is very much in line with the genetic makeup of the coral animal, because it shows us there's a homogeneous response to temperature of the coral animal. So it's it has one optimum throughout the entire Red Sea. And we already can see, okay, the corals in the south, also the corals that are in the summer, they might be, start struggling a little bit, because we can see that the growth rates are already going down. So even though they still can cope with 33 degrees warm water, it's really at the very edge of what they can do. Then we also look at the capacity for photoprotection. So that means okay, now we have excess light, essentially. How good are the corals in neutralizing the excess light? And for that, we look at the xanthophyll chlorophyll A ratio. And so what we can see here, again, is the same kind of figure. We have now temperature on the x-axis. Where's the mouse? Yeah. We have temperature here on the x-axis, and then we have the ratio here on the y-axis, and then the numbers represent the sites and the colors, the seasons. Main point here is that corals in the north are not they just have the same ratio, doesn't matter if winter or summer. But then if you go to the south, they have actually a really high capacity to regulate the amount of photoprotective pigments they have. And so photoprotective, xanthophyll is a photoprotective pigment, while chlorophyll A is the photocollecting pigment, meaning it's absorbing the light, while xanthophyll is basically neutralizing excess light, has the opposite effect. So that's why it's called photoprotective. So that's basically, the capacity for photoprotection increases in the summer. That's what we can see in all of the sites. And then, but this regulation is exceptionally high in the southern sites. Lastly here, the last parameter I want to introduce is the heat shock protein. I told you already, heat shock proteins can help corals when they are under thermal stress. So we did look at one heat shock protein, number 60, and there's 3 different sites, the 1, 2, and 5. And what we found is that in the southern side, again, they have a really high capacity to regulate the heat shock proteins this season. And at 31 degrees here, they do have a high production of heat shock proteins. So this is all the results I'm showing you now, just is to demonstrate that these corals are adjusted well to the warmer temperatures, and these are the mechanisms they have. And that's just to sum it up here, what we found in the Red Sea. So in terms of the zooxanthellae, we found there is some local adaptation through like difference in the zooxanthellae species. So that was the first slide I showed. There are different species in the north and in the south, and then in the middle. And there's this one clade, I haven't talked about the clades, but it doesn't matter. There's one species or clade that has a strong ability to regulate the heat shock proteins. Then there's another clade which we find in the most southern Red Sea, which has a really strong ability to regulate the photoprotective pigments. So it's their way to, deal with high temperatures. And we actually also do have a gene expression data, which I haven't shown today, but they also show a really strong up and down regulation of genes in the southern sites. For the coral host, so for the animal now, we found that it has a really high genotypic homogeneity, so meaning like the population in the north looks like the same as in the south, so they're all kind of the same. That's also then supported by the calcification rates we found, which is also like one curve across the entire Red Sea. It also actually has been found, like in other studies, that the Red Sea most likely has a really wide adaptation to high temperatures. So not just the corals in the south can actually deal with high temperature, also the one in the north, even though they don't need it because they don't ever see these really high summer temperatures. This being said, after I did this study, there have been a couple of major heat waves also in the Red Sea, and a lot of the corals in the south died. Actually, the reefs look devastating in the south now. Some colleagues said to me, like, you're lucky that you were there 15 years ago because you actually still saw corals and then like a thriving reef still, it's all gone now. So I really, but then the ones in the north, because they have this exceptionally high thermal tolerance, and they're not typically exposed to these high temperatures, they are actually doing really well. So they can cope temperatures that are up to 5 degrees higher than what they normally see in summer. So basically what the coral experience in the south kind of, prepares them for, or like the abilities they acquired in the southern Red Sea, and once they distribute to the north, they keep these abilities, so they are much more prepared now to deal with like hot summers and marine heat waves. Okay, so a bit about Bermuda now. So what can we do to help corals to persist in a warmer world? So people have been now thinking a lot about like what interventions can we come up with that will help corals to survive warmer waters. There are lots of like conventional reef management strategies, which are marine protected area, reef restoration efforts, but it has been found that they don't really help much in terms of like when there is a massive marine heat wave. So what else can we do? So some unconventional reef management strategies now could be, artificial shading. So shade the reef during a marine heat wave through maybe artificial clouds, or put gigantic sails, shade sails up and things like that. Or do artificial upwelling. So that means bringing colder water from the deep to the surface and cool down the surface waters. Or assisted evolution. So assisted evolution means now that's of course not manipulating the environment, but it's making the corals stronger or more thermally resistant. I want to talk about these two because that's the work I've been doing in Bermuda now. So artificial upwelling. So first of all, what's upwelling in general? So upwelling exists, many coastal areas around the world, also including Bermuda sometimes. So often it occurs, when the wind is in a certain direction along the coastline that actually pushes the water away from the coastline, which draws water up from the deep to the surface to replace the water that is being pushed out. And So that's a natural phenomenon. It's usually seasonally, or it can be driven by storm events, or in Bermuda also by eddies, mesoscale eddies. And so that is like some natural cooling. And it has been shown in many studies, if this natural upwelling happens during a marine heat wave, it minimizes thermal stress on corals, which kind of makes a lot of sense. So now we were thinking, can we do this artificially perhaps? And this is a very basic schematic, just showing you could essentially maybe pump up deeper water to the surface and cool surface waters down during a heat wave. And so this is how this could look like. It could be like a, wave-powered or solar-powered pump that is pumping up the water. So we then went ahead and actually, proposed this idea to NSF, which also funded this idea. So the last 3 or 4 years, we have now been working on this. And the major goal here was really to evaluate the efficiency of different upwelling scenarios towards mitigating coral bleaching during thermal stress, while also minimizing the risk of unwanted effects. Because it could also be, you're pumping up that deep water, it may also have harmful effects on some other organisms, or it may pump up not just cooler water, but also nutrient-rich water, or oxygen-depleted water. So all these kind of things you don't want. So we kind of were testing different depths and different durations of upwelling to kind of see what's kind of the ideal compromise. It should prevent bleaching while also minimizing unwanted side effects. And then we also investigate the longer term effects or the post-treatment effect, over a year. Does maybe upwelling, impact also recovery of the corals and the thermal tolerance within the following year. And then we also did a test on reef communities, because of course the reef is not just corals, there are many other organisms too that may respond to this upwelling scenario in different ways. This was our last team. This was team in the summer. We have lots of, it was Bermudian students, international students participating, and also collaborators. So it was a really, really great fun experience. So a bit about the first, I want to talk a bit about the first goal and the third goal here. So the first one was test the different AU scenarios and test what would be ideal. We had the different treatments. Ambient is kind of our control, 28 degrees Celsius, kind of normal summer temperature. And then we have a heat treatment, which is 30.5, and then heat plus artificial upwelling of cooler water. And this is kind of the scenarios we tested over 2 years in 23 and 24. So we imposed upwelling for 1 hour, 2 hour, 3 hour, 5, 8, and 12 hours per day into into a heat treatment. And we also then tested water from 30 meter, 60 meter, and 90 meter depths. And you can see the corresponding temperatures here. And then we tested basically how much does that mitigate bleaching. I'm only going to show you the results of the last two, of the 2024, because we actually found that all the scenarios we tested in 23 were not very efficient. Only maybe the most extreme one, 5 hours, 90 meters. So that was kind of the setup we had, the 3 different species we tested, the greater star coral, the mustard tail coral, and then the fire coral here. We broke them into pieces. Just to give you an idea how the temperature then looks like in these aquaria, you have at ambient, of course, yellow here, 28 degrees, and then red is the heated. This is over 24 hours here. And then we have the upwelling scenarios where the temperature drops down and it comes back up again, either after 8 hours or after 12 hours. And we did that whole experiment over 6 weeks. So it was running for 6 weeks, and we did that every single day. We did lots of different measurements on the corals, of course. We measured, for example, just the color. How did the color change? How did the photosynthetic integrity of the zooxanthellae change? We did incubations to measure photosynthesis, respiration, classification. And then here are some of the results. So when we look at the zooxanthellae density, so how many zooxanthellae per square centimeter of coral, we can see here. So these are the 3 different species again, like each of this box is one species. And we can see in gray, that's the very initial value, like just before the experiment. And then all the other colors are actually representing now at the end of the experiment, like after 42 days, which is 6 weeks. So, or 7 weeks actually. So yellow is ambient, 28 degrees. Red is heated, so that's the 30.5 degrees. And then the 2 blue ones are heat plus upwelling. So we can see here for the fire coral is really kind of the loser here. They're always the worst and they bleach. They're always the first one that die. They're really not very robust. Although they are very important for Bermuda reefs because they're like one of the few branching species we have. So, but we can see if you look at red, nothing, no zooxanthellae left at all inside the coral after 42 days. And also upwelling didn't really help much here, right? And I mean, if we look at the greater star coral here, we can see, okay, almost no zooxanthellae left under heated conditions, but upwelling here actually did, keep the zooxanthellae levels at least something in between ambient and heated. So that's pretty good. So it's helping to some degree. And then for the master tail coral, we can also see that the upwelling is somewhere in between, so it does have a beneficial effect. We can see a similar pattern here for photosynthetic integrity. So that's the FVFM value here. This over time now. This is at the beginning of the experiment, end of the experiment. Again, the colors correspond to the different treatments. And we can see that, again, Millipora, of course, doing very badly under heated conditions. But they actually, when we just look at the integrity, like of the remaining zooxanthellae, they're actually not too bad under upwelling scenarios. Again, here for greater star coral, upwelling did help to, keep photosynthetic integrity up. And then Porites overall didn't respond that badly to start with. And upwelling actually didn't have much of an impact here because they're not doing that bad to start with. Looking at coral growth now, there's also something we measured. Just on 2 species here, we can see that the growth also suffered, and the heat treatment. So here red is lower in both species than the ambient, like the yellow. And then upwelling did, again, help to keep the growth rates at a certain level, at least for when we upwell for 12 hours per day. So this is 12 hours per day. This is 8 hours per day. So it actually made quite a bit of a difference here. So overall, we can say artificial upwelling can mitigate thermal stress in corals. Last year's experiment was a community scale experiment. Now, of course, we know it's benefiting corals, but what's happening with all the other organisms on the reef? So we built these little communities, and I think, Amy, you've seen those, right?
Students in here is what we.
Okay, great. Yeah, I think we were just at the end of the experiment when you guys visited. And so we have here the different coral species, and we also have actually gorgonians, soft corals. So they also contain zooxanthellae. So we were thinking maybe, they should also bleach, right? Like if they have zooxanthellae as well. So we included them in the experiment. We had a lot of live rock in there, which is covered by algae, sponges, and all kinds of stuff. Then we had fish, snails, hermit crabs, and we kind of looked at different things like did the coral performance change? Did the algal community structure change? Fish behavior, how was that affected? Community metabolism, photosynthesis, respiration, recruitment of corals as well, and benthic succession. So lots of different people were involved in this, working on different topics. And I'm not going to explain the results. I'm just going to give you a couple of insights on how the communities look after the experiment, at the end of the experiment. So this was a community after 45 days at 30.5 degrees. You can see lots of white corals here. And you can also see different species react differently. And so this is a brain coral. This is a brain coral, very white. But then the master-tailed coral actually is still pretty yellow. And then the Montestraya, which is the greatest star, also really white. The Gorgonians actually, they didn't care at all. And we also, like, when we looked at the photosynthetic integrity, which is don't show the data now, but we also found that they were doing just fine. So the next step would really be like looking at what kind of zooxanthellae do they have? Like, is it a different species? Are they particularly thermally tolerant? So something we still have to do in the future. And then this one here is now from a community that was also exposed to upwelling. And upwelling for 8 hours. And we can see here that the corals are generally still have more color than the heated one I just showed before. And generally the community didn't change too much. Also the algal community didn't change too much. Fish behavior, unfortunately, like what I could say from just walking around and observing the fish didn't change much, which is also good news. But I do have to say that the butterfly fish was a little bit, they're really picky when it comes to temperature. So we did have a few butterfly fish dying in the upwelling tanks. I think they don't like the up and down of the temperature. So if we ever would do that on the reef, in reality, I mean, maybe they swim away, you know, or yeah, we have to think about if this is like the compromise we want to do. Good. So I think I'm going to skip this little video here. It's just kind of showing some of the fish swimming around. But we're running out of time. So I wanted to just very briefly mention at least the very last project here. I talked about manipulating the environment. We also do work on like trying to manipulate the coral itself. So it's more resistant or more capable of living under hot conditions. And here, basically, we assess the thermal tolerance of corals, their ability to learn thermal tolerance, and their potential to pass on acquired thermal tolerance to their babies. So if we expose the adults to heat stress, you know, are they able to pass it on to their babies, and are they maybe more thermally resistant in the future? So I'm just going to show you these results of the very last one, thermal resilience in coral offspring, so the babies. Here is how we actually get the babies. So we put the corals in these buckets here and just wait for them to release the larvae. We collect the larvae and we then add them to these kind of settlement substrates here. And you see each dot here is actually a settled larvae now. So all of them, they all like to kind of snuggle up and go together and really close. So you can see lots of them kind of being in one spot. Even a lot of them die in the first few days or first few weeks, but then some also merge and grow together into a new colony. And you can see here also this is a single polyp of a coral of a baby. So what we did in this experiment here, we took the adult colonies before we put them in the beakers here for spawning and expose them to different temperatures. Blue is what we find in the reef. So that was kind of in June. So June, of course, the temperature is a bit lower still. It's still climbing up. Then we kept it at like 28 roughly. And then we had a batch of corals that we exposed to higher temperatures, which is about 2.5 degrees warmer. And then we collected the offspring, which were released here in this gray area here during that time. And then we put them into, again, ambient and heat treatments and different treatments. And we kind of checked, okay, how well do they settle? Is there any different settlement behavior between the ones that come from heat-treated versus ambient treated coral? And actually in this figure, you can see there's not really any difference. They're all kind of the same. However, it becomes interesting in terms of survival now. So after 3 weeks, we checked, okay, how many corals are still like of the babies, how many of them survived. And what we can see here, and that's the last figure I'm showing you, I'm going to be quiet in a minute, but it's so the color indicates the parental colony, so where the babies are coming from. So in the babies here, like the parents in blue, or the blue ones means, I don't see the cursor now, The blue one means that the parent was exposed to ambient. Red was, or brown is the one exposed to heated. And then the two groups down here, you can see the group to the left is then kept at ambient, and the one to the right is kept at heated while the babies were growing. And we can see that in the heat treatment now, over here, that the babies that are coming from the heated parents have a higher survival rate than the corals that come, or the babies that come from the ambient parents. So that means like, okay, if we expose the corals to heat before they actually release their babies, they acquire some sort of thermotolerance, it seems, and that they can pass on, and then the babies are also more thermally resistant, which is good news, right? So it means there's some potential for adaptation. Yeah, and that's it. Yeah. Yeah, of course. Yeah.
I have the first one. Oh, sorry, you had the first one. I'm sorry.
No worries. I'm just curious, for the AU, how do you control the composition of your upwelling water? Do you take it from the ocean, or do you manufacture, make sure there's no nutrients, no toxins?
Mm-hmm. So in the experiment in the lab, we just cooled down surface water. So we were actually neglecting any of this, so to mimic the colder water. But we did go out and did take measurements from the different depths as well, and looked at the amount of nutrients that are in there. We looked at the carbonate chemistry, the pH, and all that. So the depths we chose were based on like the minimal changes in the chemistry, but only a change in the temperature. If you would go deeper, let's say if you would go 150 meter deep, you would definitely find much higher nutrient concentrations and also different carbonate chemistry. But because we are in the so-called euphotic zone, where there's still light and we still have photosynthesis going on, it's the chemistry of the water doesn't change too much.
So if you implemented this in the ocean, you'd have to do some sort of periodic sampling to verify?
Yes.
the source
You would, you would do that before you install it, kind of figure out what's a good depth to do this. And also, as long as you stay within the euphotic zone, like the zone that is light penetrated, it's okay, I would say. So that, yeah, it's really below, but there's no photosynthesis and only respiration going on where the chemistry changes a lot.
Cool, thank you.
No, I'll let Phoebe go. Mine wasn't as good as yours.
Again, I have a question on the artificial upwelling. So when you're doing this in your experimentation, are you just turning the upwelling on, like, and just pumping it through at the same rate? Like from start to finish, there's not like a acclimatization period of like slowly increasing that?
No, yeah, we just basically turn it on. Yeah. And it takes about half an hour until the mixing ratio is perfect. So we don't just add the cold water and like replace all the warm water. It's kind of a mixing ratio, like trying to mimic it a bit how it would be in the field. And so it's, and until this stable temperature is reached, it takes about half an hour. So they do have like a short, acclimation period. Yeah.
Yeah, and like in environmental upwellings, these would be like seasonal, like long-term things. So are you guys really looking for like what's the minimal amount of time that would be required to like actually make a difference?
Exactly. Yeah, that's exactly what it is. And you would only turn it on if there's a heat wave, right?
Yes.
You turn it on every summer.
Yeah, it'd be like an emergency.
Right. Yeah. And of course, also, you have to think of it. I mean, the reefs are huge, right? I mean, if you think about installing something like this, it's not an easy and cheap solution. So I think it's really something you can do only very localized to maybe save patches of the reef. And then those patches, well, one of the additional objectives are actually that if we cause these strong temperature fluctuations, that also has a stress hardening effect. So the corals that are protected within these little patches of cooler water are hopefully also getting more thermally resistant that then seed, damaged reef areas. Yeah, so that's kind of the idea. We're still, working at this project is still ongoing. We will test them again this summer if they actually acquired some thermal tolerance, like the corals that have been exposed to upwelling. And so we'll see. It's still ongoing. I don't know. Okay, Robbie, we only got 30 seconds.
Excellent. That was great. Were you able to look at changes in the diversity of the zooxanthellae clades in the AU treated colonies to see whether there was a shift, some not being beneficial?
Not yet, but we have the samples in the freezer, yeah, for that
Get
purpose.
the answer to my question?
Yeah, good.
I had a question about the artificial upwelling as well. I'm sure you've considered microplastics. So how do you dodge repolluting and keep microplastics from being recycled into the environment because they settle on the floors? So how do you keep that from?
Good question. I have not considered that. But I think where we take the water, it wouldn't be necessarily from the bottom. It would be a bit further up. And then I don't think we can prevent it, to be quite honest. Yeah. So it's, I don't know too much about the accumulation of microplastics. Is there more in like 90-meter depths than there would be in 10-meter depths? I don't know. So I think, but something that maybe should be considered, you know, before installing something like this, look at potential pollutants as well.
If it's a cost thing, would you consider the shading as your next project?
The shading, yes, actually, I've been talking to colleagues about that as well. It would also just be a very localized solution, of course. I'm not a big fan of creating clouds and stuff. It just seems a little crazy to me. But it's also maybe, yeah, I mean, you also have to release something into the air that might be harmful for the atmosphere, I'm not sure. But you could, you know, what you could do is underwater create like shading tables. And then again, you can just make it very localized and maybe have a mosaic of shades and hope that at least you know, are able to save some reefs or some corals that are then able to reseed other areas.
In your Red Sea study, you mentioned the, I believe, a modification internal in the coral to actually change its shading to an internal shading of the sunlight coming through. I don't know if it was reference to chlorophyll A, but is there a color change in the coral? How is the coral actually carrying this out?
I think, I'm not sure if you would visually see a color change, but it can. Usually when we see a color change in a coral that is seasonal, it's more like they get more pigments when there's less light because they need more pigments to harvest the same amount of light. So typically in winter, we would have slightly darker corals than in the summer. But I don't think you would visually see a change in the photoprotective pigments, but I'm not 100% sure. Yeah.
I just wondering if this was a survival characteristic that was occurring.
Yeah, it definitely is. I mean, they have like, these are just the two of the pigments I showed. They have more pigments than that, and some are protective and other are photo harvesting. So it's kind of like, and they can up and down regulate them as needed. Yeah.
Have you explored this? It's got to be some good correlation. Have you explored, without releasing unknown things into the Bermuda habitat, in trying to identify or develop the kind of genetic diversity you saw in the Red Sea in Bermuda?
If that's something we would be striving for, I don't think it will work, because what's really limiting in Bermuda is the cold winter temperatures. There are not many corals that can survive the cold winters. And I mean, it's already, and also because we are connected to the Caribbean in a way, and the Caribbean has way less, and also has way less coral species than the Indo-Pacific. So we naturally have lower diversity, and then it's really the environment that determines the diversity in Bermuda.
We can't have it all.
Yeah, no, can't have it all. But we have the really hardy ones, so that's good. Yeah. Anything else? Sorry.
Does the Gulf Stream actually help in the artificial upwelling? I mean, Bermuda's right in the middle of the Gulf Stream, right?
Yeah, we actually a bit south of the Gulf Stream, but not directly. But what happens with the Gulf Stream is like the Gulf Stream passes Bermuda to the north, and then there are lots of eddies, mesoscale eddies that spin off, and those eddies can create upwelling. So it's not coastal upwelling then, it's actually upwelling in the middle of the ocean, but those eddies move, and then when they hit the reef, or they could essentially, cause upwelling in the reefs as well. And I have another NSF project that is looking exactly at the impact of mesoscale eddies on reefs. We have not been very lucky with this project because they just never occur when we want them to occur. And they do occur more in the winter, but then in the winter it's not really relevant because the water column is mixed so well anyway from all the storms and stuff. So when it would actually make a difference in the summer, we haven't really been able to catch them, essentially.
Is that the eddies from the Gulf Stream? Is that what keeps the coral in Bermuda alive in the winter? Because it's coming from the south, it's bringing the warm water up.
Absolutely. Yeah, that's right.
That's why it's surviving.
Yeah, exactly. Because otherwise, yeah, this latitude would be colder if you wouldn't have the Gulf Stream. Yeah, that's right. It's the same actually in the Pacific too, like Japan, the southern part of Japan is about 32 degrees as well, like north. And they have I've forgotten how the current is called there, but it's the same. Which one? Horatio current. Yeah, it's also this warm water current that's coming up, from the tropics and up and bringing the warm water. Yeah.
Are there any strategies being employed in this part of the Red Sea to try and address the loss of?
There is definitely research going on, but I'm not sure exactly how much they do in terms of actual trying to combat also it's in areas that are logistically not easy to accessible. So I'm not really aware of. They do some research on trying to also create thermally more resistant, thermally resistant corals, but it's a long process. So it's at the moment, I don't think there's a lot they do actively against losing those corals, just because it's not possible. They just don't know how. Yeah. And it has been happening so fast. Yeah.
And what about in the Caribbean?
Yeah, in the Caribbean, there's lots and lots of research going on in also creating thermally tolerant genotypes and species. And but the geoengineering approaches I have mentioned, like the shading and the upwelling, is not something that's really done yet. There are some pilot studies, but it's not done on scale that it would have any serious impact, unfortunately. There's quite a bit of research going on in terms of shading, in using cloud brightening and also some other tools in the Great Barrier Reef in Australia. Yeah, because they're also massive loss of reefs due to marine heat waves.
So Bermuda's a bright spot in all of this.
Yes. Yes, it is.
Your slide at the first mentioned seagrass, and I know the turtle and seagrass and the growing. Could you address just for a moment what your research is showing on seagrass?
Yeah, so it's a few years ago now. I was working with DNR on this project, the Department of Environment and Natural Resources, and what we found is generally that, you maybe know that a lot of the seagrass has gone in Bermuda, and people have been blaming the turtles for it, more or less, and they definitely have, they play their role. But of course, it's also because human impacts already reduced the amount of seagrass meadows, so there was not as much left for the turtles, and then the turtles are the one that kind of do the rest essentially. I have seen, just from not doing research but just from observations, that seagrass is coming back in some areas. Robbie may actually know more about this, but it's coming back in areas, and just in front of my house, actually, I'm always excited going snorkeling and seeing that seagrass coming back, and turtles, lots of turtles actually there as well now. I don't know how well the cages worked that have been put out. Not so much in the end, right? Yeah. So at the end of the day, we just kind of have to let it go. It's natural, the natural thing. Like what we try to do in terms of caging, by keeping the turtles out of the seagrass. And we also tried some seeding methods, like seeding seagrass, which was also not very successful. But now it's good to see like naturally seagrass is coming back.
Why was the seeding not successful?
Do you know
What was
why the seeding was not successful?
We're still trying to figure that out.
Yeah.
In terms of the fact that, you can put a bunch of carrot seeds out and you get a percentage that comes up and you're happy with that. It's just other factors go into making that seed successful at that particular place or experiment you put them in there. They just don't seem to be very robust at germinating because for them, their success is to just grow vegetatively rather than occasionally have a seed.
Exactly. Yeah.
Unfortunately, where's the case our restoration work then. So. But there's, as I'll just support Yvonne's assessment here, that seagrass is coming back in places where we saw it decline and recover. But we've learned from the Bermuda Zoological Society and the Aquarium's turtle project, the turtles are just more broadly spread over. Since they had nothing to consume close to shore, they had to go get something to eat somewhere. They're out on the reef. I see them out there frequently feeding on algae, other things. We look at some gut contents and they're feeding on all sorts of different things in that. So an adjustment, and we've seen this happen before in the past. Seagrasses come and go, turtles come and go. We need to do more to allow seagrass to be successful. That's tidying up our inshore waters in some ways to reduce boat moorings, shift that process around. We still need turtles. They're still useful. They're still protected. They're still important. We just have work to do. And perhaps we'll get lucky with some restoration work. We have a Bermudian student, Triana Zool, busy with her PhD down in Florida, learning how to do more seed propagation work. Hopefully she'll bring some of that skill and knowledge back up to accelerate this process.
Does ocean temperature rise? Is that beneficial for seagrass or harmful?
Seagrass is pretty tolerant, generally speaking. Everything happens.
Yeah, especially it's also a tropical species. With all tropical species we have here, that turtle grass, manatee grass. So I would think they're probably okay with a couple of degrees. Of course, it's because I haven't tested. So, yeah.
Is there something unique about the area that you're seeing the seagrass increasing that you might think, that's less marine traffic, more inshore?
Yeah, it's like it's Mullet Bay area in St. George's. Yeah, and well, it's a very protected bay. I mean, there's definitely some moorings, and you can see where the moorings are. There's no seagrass, right? So you can see the circle where the mooring chain is going. But everywhere else, we have the manatee grass coming back. And that's kind of more a pioneer species. So that's one of the earlier species. And then the hope, of course, is that once the manatee grass is established, then the turtle grass will come back as well. So the turtle grass is a thick, wide-leaved grass. Yeah, so I'm not exactly sure why they're successful right there. I think it's just, yeah. As I said, I haven't really kept studying the seagrass. I haven't really done comparisons. But it seems like because it's protected, because one of the problems is if seagrass is decimated so much that the roots are gone as well, then all the sediment, kind of sloshes around, and there's no stable substrate anymore. And then it's really hard for new seagrass to anchor themselves into the ground and grow. So, but if there's something in place already that keeps the substrate stable, then it's easier for new species to come in and grow.
Because there's a lot of abandoned boats, sunken boats in Bermuda that maybe slosh around down there. It'd be interesting to see if there's any observable around those sunken boats. There's a project KBB is heading up to get the abandoned sunken boats out of the ocean.
Yeah. I mean, anything that's sloshing around on the bottom, I think it's not gonna. Yeah, it's gonna be kill the seagrass essentially. Yeah.
Risk of asking a stupid question because I really don't understand a great deal, but is there an opportunity to generate or to create a more resilient coral that can then be used in transplanting where corals are damaged? Is that a fair question?
Absolutely, no, absolutely, and that's exactly what people are trying to do. Yeah, they try to do it either by transplanting corals that are already naturally grow into more harsh conditions and then into environments that are becoming harsh, so, or you can do that in the lab too. Yes, you can create. And that's kind of the last project. I was a little short now, but that's what we're trying to do. We kind of, stress the adults to some degree, and then hoping that the offspring they produce are more thermally tolerant. And of course, we could use those offspring and plant them back to the reef. So we actually have now. That's the Heising Simons Foundation project, which just stopped, but we're now going into the next phase, third phase, and we will keep working with the same corals and see over time, over generations now also, if we can increase their thermal tolerance. Corals grow really slow, so with these projects, you always need a lot of patience. But yeah, we are definitely working on this, and not just here, but many other places. In particular, the Caribbean people work a lot on this question as well.
Sorry, can we make you the last question? Because I think
sure.
there's a lecture.
Yes, I had.
You got a few minutes, but yeah.
I noticed earlier in the slides in the Red Sea that the biodiversity of the bacteria dropped a lot, and then it seemed that that's.
I'm not sure if the diversity dropped or if it's just a community change.
Okay.
Like the bacterial.
So I was just wondering what it's like in Bermuda. Like what is the biodiversity like in Bermuda?
In terms of microbes?
Yeah.
I don't know. So we actually have my previous postdoc who just finished his position in my lab, but he keeps working with the data. He did a study on the microbes, but he did it not in the reef. Well, he took some samples in the reef, but then he also took mostly the microbiome from the corals that we kept in the from the experiment. And he's looking at the change in the microbiome on heated versus ambient corals, but also adults versus offspring. And then compare it also with what we see in the reef. Yeah. So I know he just got the data back and he's working it up, but it will still take a bit of time until we have the results. Yeah. Okay.
All right,
Well, thank you so much. That was fun.
Real quick, just to let you know, if you're around in a month's time, we have another one. So on the 26th of March, same time, same place. And then again in April, will be our last semester. And just to say thanks to Chubb, they were our sponsor for the thanks to Baz, and thank you to Dr. Sawall. Thank you, everybody. Thank you.
I could hear the text.